So just to review a little bit here. So enzymes, also called biological catalysts, they're made by living things. They catalyze or speed up chemical reactions by lowering the activation energy. The molecules that the enzymes work on, the enzymes catalyze, are called the substrate molecule or molecules. So in this example, we have an enzyme, which is here in pink. It's working on these substrate molecules. The area where they all sort of bind together is called the active site. And once they bond together, they form this enzyme substrate complex. And then there's some change in the substrate molecules. In this case, what did this enzyme do to these substrate molecules? What would the term for that? Yeah, this is synthesis. It's the opposite of digestion. Right? Digestion is to take a larger molecule, break it into smaller molecules. This enzyme is a synthesizing enzyme. It's taking smaller molecules and allowing them to bond together. And so the result is called the product of the reaction. The enzyme, however, is not changed. It can be reused over and over again. So once the products are released, new substrate can connect with the enzyme, and so it can work over and over again. So, oops. So there's a couple, so if you notice on the diagrams you've seen so far, the enzyme and substrate are always drawn sort of with like a complex shape. They look like puzzle pieces sort of fitting together. That was sort of the original, when scientists first started identifying enzymes and figuring out how they work, they developed what was called the lock and key model. This shows that lock and key model. What they originally thought was that the shapes must all match up perfectly, kind of like the pieces of a puzzle. And once they all connect, then the reaction takes place. Later on, however, in like the middle of the 20th century, scientists sort of changed their model of how enzymes work and started thinking about this as this induced fit model. And what the induced fit model, how it's different from the lock and key, is that, as you can see, this shows the induced fit. What happens is the substrate, it's not a perfect fit with the enzyme. It's not like a puzzle piece here. The shapes are sort of roughly similar. Okay? They fit in there sort of loosely. But then once the substrate molecules actually bind with the enzyme changes its shape of the whole complex, and then the reaction can take place. So that was a sort of change in the theory of how enzymes work that happened from the beginning of that research till what we think today. So when we see them here drawn as a puzzle, it kind of helps us to visualize, helps us think about the enzyme fits with the substrate in some way, but it's not perfect like this. It's not a puzzle piece that clicks together. It's kind of a rough fit, and then everything binds together. So if we think, here are some examples of human enzymes. We often think about enzymes in relationship to digestion, but there's other types of enzymes that do other work, but these are digestive enzymes. And we have a whole variety of them. Remember, an enzyme is specific. An enzyme only works on one specific molecule because the shape of the molecule, the substrates, matches with the shapes of the enzyme. Just remind me though, enzymes are made, what type of molecule are they? Proteins. So a protein is a large polymer, a large molecule made of smaller repeating units. What are the repeating units for a protein?
Carbon?
Nope. Just say words. So a protein is made of a bunch of these linked together.
Mitochondria.
No?
Oh.
There are 20 of these that can possibly go into making of the protein.
Oh, amino acids.
Yes, amino acids. So stringing together hundreds, thousands of amino acids creates one protein. So a protein would be made of thousands of these amino acids. All right, so
I could jump tomorrow.
one example, protease is an enzyme. Remember, enzymes often end in ASE as the end of their name. So protease is an enzyme that breaks down proteins into amino acids. And these are found in the stomach, proteases. In the small intestine, there are sucrase enzymes. These are enzymes that break down sucrose into its monosaccharide components. There's also lipase, which breaks down lipids into glycerol and fatty acids. In our mouth, saliva contains salivary amylase, an enzyme that breaks down starches into disaccharides or monosaccharides. So different enzymes work on different molecules. They're specific. Several factors can affect how an enzyme, how active an enzyme is. One of those is temperature. We look at this graph in the lower left. So the y-axis shows how the enzyme activity, how the level of enzyme activity. The x-axis here shows temperature. So at very low temperatures, these enzymes are not very active. As temperature rises, however, they become more and more active until they reach their peak. The peak, when that enzyme is working the best, is called its optimum temperature. What happens when you increase temperature beyond that?
Probably decreases.
They decrease eventually and then eventually stop working at all. So once the optimum conditions are no longer present, then the enzymes don't work as well. This is one of the reasons that having a really high fever can be dangerous. What temperature do these enzymes work best at? Yeah, right around 40, 37. That's human body temperature. So when the person gets a fever that pushes their body temperature up to 40, 41, 42, well, these enzymes start to decrease in how well they're working. And that interferes with how our body works. If you heat up an enzyme, even beyond the point when it stops working, it can eventually become denatured. What that means is the shape is permanently altered, and the enzyme will no longer work. Okay? It will no longer function with the substrate molecules if it's been heated up to too high of a temperature.
What can cause that to happen?
In biology, people's body temperature doesn't reach those levels to denature the enzymes. But for example, I don't know if I should tell you this yet. I'm not going to tell you because we're going to be doing a lab when you're going to explore temperature or pH effects on enzymes. So I'm not going to say too much yet. I will tell you more once we do that. Griffin?
Why do grapes explode in the
I
microwave?
don't know.
What is the level of temperature?
Human body temperature is 37 degrees Celsius, 98.6 Fahrenheit. That's average.
No, like what is the max?
Stuff.
Oh, I mean, once you get to 105, 106, like that could be fatal. Yeah. All right. pH also influences how enzymes work. If you look at this middle graph, here we see pH on the x-axis going from 0 to 12 or whatever. And then on the y-axis, again, we have enzyme activity. And what you'll notice is that these different enzymes, pepsin, salivary amylase, alkaline phosphatase, each of them has a different optimum pH. Like, what is the optimum pH for pepsin?
1.5.
Where do you think you would find pepsin in the human body?
Probably your stomach.
Go through.
Yeah, in the stomach, because the stomach has hydrochloric acid, very low pH conditions. Pepsin works best in those conditions. Pepsin is found mostly in the stomach. Salivary amylase is found in the mouth, which has a relatively neutral pH. Alkaline phosphatase is found in the small intestine, which is slightly basic. And again, if we look at its optimum pH, it's a slightly basic pH. And so if we change the pH lower or higher than the optimum, that also affects the enzyme activity. Griffin?
What exactly does basic do like a substance?
Excuse me?
Like, you know how acid disintegrates stuff? What is that?
I mean, acid, both of them can be damaging to,
So it's an
material.
animal.
So a strong acid or a strong base, both can be equally dangerous. The other factor that can affect how well enzymes work, the rate of enzyme activity, is the concentration of the substrate. So if you imagine, if you had a lot of the enzyme but only a small amount of the substrate, the enzyme action will be relatively slow. But as you add more of the substrate, enzyme activity increase and increase until a certain point. Because at a certain point, all of the enzyme molecules are occupied by substrate. And therefore, adding more substrate isn't going to increase the reaction anymore. We say it's been saturated, and that's what this green X is, showing that we add more substrate, increases it until a point, and then it's saturated, adding more is not going to do it. And the last slide for today. So some medications, some drugs are, they work by mimicking substrate molecules. One example is a really important drug. The penicillin family of antibiotics are called sulfa drugs, used to treat bacterial infections. And the way that these sulfa drugs work is the structure of the molecule which is sulfonamide, is very similar to the substrate found in bacteria called PABA. And bacteria need this compound PABA in order to make folic acid, otherwise they can't survive. So these sulfa drugs mimic this PABA molecule, and they occupy the enzymes rather than the PABA, and therefore the bacteria cannot make the folic acid and eventually die. So some drugs work by mimicking the shape of substrate molecules. And you'll see this is a common way that medicines work. It also works not only with enzymes, but receptor molecules and neurotransmitters. Many drugs work by sort of mimicking similar drugs actually found in the human body. Any questions about enzymes? All right.